My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. I am here with Eva Kerr and Jade Samples, and we are going to have an interesting conversation today. We're not going to talk about snakes so much, although I bet they have a lot of great snake experiences, and they might even have a snake story up their sleeves. But we are going to talk about pond breeding amphibians. And in another life, I used to do and still do quite a bit of work with pond breeding amphibians. My master's work way back when was on vernal pools and salamanders. And so this has always been a kind of a group of animals and that are associated with a particular habitat that have been of interest to me. And this is part of our student perspectives series. So both Jade and Eva just recently finished, recently within a week ago or something like that. So I'll say congratulations to both of you and welcome to the podcast.
Thank you. Happy to be here.
Good. Well, if you've heard any episodes, you know I like to hear the origin story. People think we're strange in that, we like snakes and salamanders and frogs. And I'm curious to hear how both of you got into these things. So we'll start with you, Jade. How did you get into herpetology, amphibians, snakes, reptiles, these types of things? Is this something you've always been into or something kind of more recent?
Yeah, so I grew up in rural Alabama, and so I was always playing in the woods, kind of the classic story you hear from a lot of wildlife people, as they were just in and around and with nature a lot. And I just knew that I liked animals in general. I did a lot of catching toads outside my house. And so when you do that, everybody says, "Oh, that's awesome. You love animals. You're going to be a veterinarian." And so I went into college on the pre-vet track. And it wasn't until my second year of college that I met someone who was studying wildlife. And I kind of explained my interest to them, and they were like, "Well, why are you not studying wildlife?" And I said, "Oh, I didn't even know that was an option." And so I got into the forestry and natural resources program. And part of that degree program is you have to take all the ologies. So you have to take mammalogy, vertebrate, natural history, ornithology, herpetology. And I ended up taking herpetology one of my first semesters in that program, and I pretty much never looked back. I got a herpetology position, a field-based position right out of undergrad, and I just kept finding myself drawn back to those positions. I wouldn't consider myself to be a quote-unquote herper, but I am someone who considers myself to be a researcher that loves to work with herps and study herps and their systems. And so I think I found it later than a lot of people found it. But once I found it, I feel like nothing else has been the same. It doesn't scratch that same itch.
Yeah. Yeah, I'm the same way. I'm not much. I won't say on occasion I'll go out and look for reptiles and amphibians recreationally, but that's not my primary form of recreation. It's my work, and I'm fully dedicated and fascinated. But yeah. So you grew up in Alabama, rural Alabama, and then I didn't catch where you went to undergrad, but did you go to Auburn?
Oh, yeah. I did not go to Auburn. Blasphemous in my family, but I did go to UGA. So I went to the University of Georgia, and I was in the Warnell School of Forestry.
You're a smart woman. Go dogs. Anyways, so you went to University of Georgia, and Where's just continuing that side tangent, was your family an Auburn family or an Alabama family?
They're actually a Georgia Tech family, so it's even
Oh, okay.
worse that I went to
Okay.
UGA. But it's funny, I
Gotcha.
got to college and realized that that is a one-sided kind of hate relationship.
Yeah.
Because when I came to UGA, everybody was like, "Oh, ew, Georgia Tech, okay."
Yeah. Gotcha. Okay. So you get into college, and so it kind of hit you. You love nature. You're experiencing it, but but you're experiencing all of it. It's not just herps. And then, like you said, you just kind of dove full in into reptiles and amphibians. Well, I'm going to wait on that. Let's go over to Eva. And Eva, how did you get into these amazing animals?
I have a relatively similar background. I grew up in a family that encouraged spending time outside and just enjoying nature. And I always remember like my dad moving snakes or snapping turtles across the road, and just grew up with a lot of experiences like that. And my parents sent me to a summer camp growing up called Nature Camp. And you had majors while you were there, and one of the majors was herpetology. And so I just think from a very young age, I was into it. I really haven't worked with any other system besides amphibians and reptiles. I grew up in Virginia, and then I went to undergrad at a really small school in Pennsylvania. And I had pressure sort of to be an engineer because I was a good student in high school. But I really quickly switched to being an environmental science and biology major, but I kept my math minor. But even though I went to a small school, I got a lot of connections and experiences. So I ran cross country for my school, and one of my captains and someone I really looked up to begged all of us for help on her senior thesis. And I went out and helped, and it was on the spotted salamander migration, which is a super easy way to fall in love with working with amphibians. Nothing like holding a bucket full of spotted salamanders. And then that next summer, I spent in the Rocky Mountains working on Arizona tiger salamanders with Dr. Howard Whiteman, which was super cool and just showed me about having a career in research. And the next summer, I worked a similar job in Arizona on Arizona tree frogs with a grad student. And then I was like, I definitely have to go to grad school and pursue this. So I had a relatively straightforward path to it.
Gotcha. So you're kind of always into them. You were majoring in herpetology when you were young. So how old were you when you were doing that, the camp experience there with the herpetology major?
Probably like 12.
Gotcha. Okay. Interesting. So, but you both ended up in graduate school at University of Georgia. But again, you're at UGA, but you're also kind of affiliated with this other site that's tightly affiliated with a school named HOA or the Jones Center, and we've interviewed Laura Smith, who's the herpetologist that I'm sure you both know very well. She's been on previous episode, and so how did that happen? I mean, Jade, you were already at UGA. Did you kind of build connections while you were there during your undergrad? Were you already connecting to labs? And but basically, how did you end up meeting Laura? How did you end up getting connected to HOA and transitioning into a master's program at the same university that you're already at?
Yeah, that's a great question. I actually graduated kind of in the COVID era. And so it was a little bit difficult in undergraduate to get those research opportunities as it is kind of in a normal year. And so I really tried to reach out and volunteer on any projects I could or work in any labs that were still functioning. And so I got a little bit of undergraduate experience. And then I mentioned I got a herpetology position right out of undergrad. It was working on plethodonid salamanders in southern Appalachia. And so I worked that position and I worked a few jobs in between there. I worked on the Gopher tortoise crew for Georgia Department of Natural Resources, and then came back for the second season as the crew leader for the salamander position. And the PI on the project was Dr. John Mayers. And I had been talking to him about my interest and that I was really thinking about grad school. I had done number of technician positions, and I'd been a crew lead a couple times, and I really felt that there was not a lot of upward mobility for me, and I had kind of had enough time to reset from COVID education, and really was looking forward to being back in school. And so he kind of sat on that for a few months, and then it just so happened that a grant came through from Georgia Department of Natural Resources, where there was some funding for a master's student to come on and look at some of the wetland restoration they had done for amphibians. And so he reached out to the Jones Center and Laura, and then the Jones Center was able to hire me as a technician for 7 months before I started grad school. So I actually was able to work as a technician and collect data during that time, and then start in August of that following year. And so I met Laura as a technician down there who was leading into a master's position, and she was my co-advisor for my master's. And so her and Dr. John Mayers were my co-advisors. And it was a wonderful balance to have. They kind of, I like to say they like to even each other out in a lot of aspects.
Mm-hmm. Yeah, I can see that, knowing them both very well. So where were you when were you working out of Coweta area when you were up here then?
I was working out of a lot of spots. We had about 28 field sites. Most of them were in Coweta. A lot of them were in Great Smoky Mountains National Park. A lot were up near that western North Carolina, Tennessee border. And we were just trying to get a huge gradient of climate and elevation. And we also had some sites near Unicoi.
Yeah. Okay. That's right where we're based. That's right where I live in that area. So know it very well. How about you, Eva? You have a different story, I'm sure. You weren't, it sounds like Jade, you stayed at the same university and you probably used some of the connections you had built, but you ended up working quite a bit in between and building a lot of connections. But Eva, you were coming from a completely different place. You were coming from Pennsylvania, having, some of your first real field experiences, it sounds like, down in the desert Southwest. How did you make that transition to ending up in Georgia?
Yeah, I actually entered grad school straight from undergrad. So my senior year of undergrad, I was cold emailing a ton of different people. But one of my professors at my university knew Dr. Mayers. And so that helped. And then the PI I worked under in Arizona also knew Dr. Mayers really well. And so they helped me connect with him. And I really only ended up applying to UGA. And from there, it just was a dream come true. And I started at the same time as Jade, so in the fall of 2023. But I didn't really come into a project. There wasn't a grant or anything that I was coming into. So I sort of got to build my project when I came.
Gotcha. Yeah. So, I think that illustrates for anyone in the audience who's moving into this career, just the importance of, it's like this with most things in life, right? But the importance of networks and who you know and connections and building those networks over time are just absolutely critical for success. And there's some of that kind of baked into both your stories there. So you're both at UGA working again between Dr. Mayers and Dr. Smith, and your field work, I'm assuming you're living in Athens, but your field work is down in Southwest Georgia at the Itchaway site. So I guess before we start breaking down what each of you have been working on, maybe one of you or both of you could give us kind of like an overview about Itchaway. Again, they can go back and listen to that episode with Laura, but just give us a real high level overview about what it is and a little bit maybe about kind of the landscape there and especially those factors that might be real important to your thesis projects.
I can do that, Eva. So Itchaway is a research facility that is based in Southwest Georgia on the Dirty Plain. And so for us, that's really important because all that karst limestone topography creates depressional isolated wetlands. And those wetlands are what a lot of priority pond breeding amphibians live in. Ichway is a really special site because it's kind of been managed in an ideal or desired condition since the early 1900s. It used to be owned by Robert Woodruff, who was a former CEO of the Coca-Cola Foundation. And then in the 1990s, I believe, it was converted into a research facility. And so that's what the Jones Center at Ichway is now. And there's a bunch of different departments within it, but really it's been maintained and prescribed fire either annually or biannually for a very long time. And it has this huge second growth longleaf pine forest on it that has provided a really unique habitat to do any types of studies on. And I hope I didn't butcher any of that history of Ichway.
No, I think that's good. When you kind of mentioned this when you talked about the karst topography, but one thing I always thought was really interesting and is obviously going to really come into play when we talk about your theses is just the amount, the density of isolated wetlands on parts of that property. There's a lot of ponds and, vernal, ephemeral type wetlands and all of that out there, which makes it a pretty interesting landscape from an amphibian's perspective. So when you came into your project, so you mentioned, Eva, that you didn't have a set project. How about with yours, Jade? Did you come in and there was an existing project? You mentioned the wetlands restoration, so that part of your thesis was kind of set. Is that right? And
yeah.
And so, Eva, so what we'll get to is that the two of you ended up, your work, ended up overlapping quite a bit. I'm assuming you guys both kind of did field work on both of your projects together. And so did you kind of develop your thesis ideas around the existing grant project, Eva, meaning do that on purpose, working in the same landscape so the two of you would overlap and have that type of synergy there? How did that come together, Eva?
Yeah, it was very intentional. Dr. Mears does a really good job of sort of pairing up grad students. Jade and I accomplished a lot of field work, and it was because we were able to divide and conquer. And so while we have Ichway, we also have sites on the other side of Georgia towards the coast. And so I tackled a lot of our sites on the coast while Jade tackled Ichway. And so my project was really born out of one of our sites on the coast, where we have gopher frogs sort of existing on distinct sandhills. Where on Ichway, it's more of a cohesive sandhill. Rather on the coast, there's tidally influenced streams and stuff like that. So we started to get interested in what landscape features are driving where gopher frogs are.
Gotcha. So you kind of had some leeway to develop it, but you're kind of working as a team with Jade, both in that the work you do in Ichway and then also working in separate landscapes as well. So you both could be gathering data. So you worked primarily on gopher frogs, and then Jade, I know you worked on kind of like a whole suite of species, and then also had some focus on the newts and these restoration projects. So was your whole project kind of generally conceived ahead of time through this grant? Were there parts of it that you had the freedom to kind of develop? How did that all come together?
So a large part of my project was based on the grant. We have to satisfy certain deliverables that are required by the grant. And so for us, that was two priorities. That was focusing on how varying wetland conditions and states affected all amphibian species. And so we ended up detecting 28 amphibian species for the project. And then the second priority was how it affected species of greatest conservation need. And so for us, we considered those to be gopher frogs, striped newts, tiger salamanders, and ornate chorus frogs. We did not have any flatwood salamander detections, but if we did, that would have been one of them. And so all of our sites, we had 5 different sites, including the Jones Center, had either current gopher frog populations or historic gopher frog populations. And so that's kind of where Eva came in.
So you ended up working not just on the Jones Center as well. You were on Itchaway.
Yes.
You were working maybe in these coastal wetlands and, kind of, I'm assuming, across the coastal plain of Georgia.
Yes, across the entire coastal plain of Georgia. My project did 55 wetlands that we monitored for since 2019 to 2024.
Gotcha.
A massive data set.
Yeah. Well, let's start with some of that. Let's just call it the more broad scale surveying and like the occupancy modeling. And then I want to dive in a little more in depth and talk specifically about the gopher frogs with you, Eva. And then we'll come back, Jade, and we'll talk about wetlands restoration and the newts. Does that sound like a good cadence there? Perfect.
Sounds great.
So you've already mentioned, Jade, you've got all these wetlands across South Georgia. Just talk a little bit about, just kind of how your project was structured, what you were trying to determine. We've used the word occupancy. Some of the listeners will understand what that means, but you might even get a little more specific with that. Were you looking for certain species? Were you looking for all species? Just kind of start into that part of your project and we'll kind of see where the conversation takes us.
Yeah. So originally the grant was funded to do certain restorations on geographically isolated wetlands. And we just use that to mean any wetlands that don't have any inflow or outflow of water. They're completely surrounded by upland habitat. And those are really important for a lot of our pond breeding amphibians because they're typically fishless wetlands. And when conditions are correct, you get these really beautiful open canopies and emergent grassy herbaceous vegetation. And so that is key to breeding habitat, but also foraging and refugia for pond breeding amphibians. And then a lot of that has been degraded since colonization and fire suppression. And so we see a lot of canopy closure. So most wetlands, when the canopy closes, you'll lose the hydro period, you'll lose that grassy herbaceous layer. You'll get a buildup of duff and leaf litter in the wetland basin, which makes burning even harder. And so a lot of the restorations look like canopy thinning, either mechanical or chemical. So like hack and squirt, where you put some glyphosate on some trees that you cut down. And then they also have done a few things like ditch plugging in certain wetlands. So a lot of these wetlands have been ditched for timber or pine stands to help drain so it doesn't kill the pine trees. And so there's a few wetlands that we had in the study where they had actually experimentally plugged those ditches to see if it would help recover some of its natural hydrology. And so the original goal of the project was to pair a bunch of restored wetlands and unrestored wetlands, as well as itchaways, which we considered to be in historic or ideal conditions, and look at how the presence of amphibians varied. And so we talk about occupancy, and occupancy just means, are they there or not? We don't really get a good count. We don't have abundance data because of a lot of the types of methodologies that we used, but having an understanding of how many wetlands are occupied on a site and whether or not any are occupied by certain species is a really good indicator of how things are going, especially our specialist species, like those priority species.
Hey everybody, I just wanted to take a quick break and tell you that the Oriane Society is dedicated to protecting some of North America's most at-risk species and their habitats, like eastern indigo snakes in the longleaf savannas, eastern hellbenders in the Appalachian highlands, and wood turtles in the great northern forests. Healthy ecosystems depend on these incredible animals, but these landscapes and species need our help to survive. If you care about reptiles, amphibians, and the wild places they call home, visit www.orien.org to learn more about our work and how you can get involved.
So you've got all these wetlands distributed across the state, and you're trying to figure out if these amphibians occur there. What was kind of like the technique? Like, how did you survey for these amphibians? Obviously, there's salamanders, there's frogs, there's all kinds of different techniques relative to calling and visual surveys, and terrestrial techniques for catching amphibians. How did you effectively sample across all these different species in so many different types of wetlands?
So we had two different types of methodologies, and both are imperfect types of methodologies. We had dip net surveys, and so that was targeting larval amphibians and salamanders, and sometimes we would hear calling adults, and we would also record those during those surveys. And we had acoustic recording units out in the wetlands. And so those would be out in the wetlands and record 5 minutes on the hour, every hour from 6 p.m. till 2 a.m. And those are targeting your calling frogs, your target calling adult anurans. And so we manually listened to all of those frog call files. I think we ended up listening to 31 days worth of frog call files.
Wow. It's impressive.
By ear. Yes, UGA is going to have to pay for my hearing aids later.
Don't they have software that will do that? Shouldn't AI be able to help us with this issue?
And, I keep saying that now that I'm graduated, somebody's going to come out next month and be like, we did it. It's perfect. It's just like the Merlin app. Yeah. But we have looked into it a couple of times, and there's a few softwares that claim they can do it, but not as reliably as just doing it manually, especially at places like the Jones Center where you can have 13 species calling at once. Mm-hmm. The computers just have a really hard time being able to distinguish calls when there's so many overlapping.
Gotcha. So you're doing these dip net surveys, you're doing these audio, frog logger, we'll call them type surveys. And that's how you're determining if the species are there. And then, occupancy, I'm saying this for the listeners, and correct me if I'm wrong, but in most cases, there's some unit space, and you're kind of presenting occupancy almost as like a proportion, like a percent chance that a species occupies a wetland, and you're incorporating our ability to detect them into that. And we don't need to go into that in depth, but like, what were some of the major patterns that you saw in terms of amphibian occupancy? And I mean, these can just be real descriptive. Like, we found, 30 something species. Like, this is an amazing superlative, but then, also relative to your different, the categories you described, restored wetlands and, the whatever, like kind of these historic condition wetlands on Ichaway. But what did you find basically with your occupancy results?
Yeah, so something that was really powerful about our occupancy model that we created is that it's an integrated occupancy model. So we took those two different types of methodologies and were able to force them to agree upon a shared probability of occupancy. And that's kind of a oversimplified version of how an integrated occupancy model works. But essentially, you're just able to do two methodologies to get one estimate of occupancy. So how likely they are to actually be in those wetlands. And some of the specific things that we were looking at were how does the hydroperiod affect them? How does distance to the nearest occupied wetland affect them? And how does canopy cover affect them, as well as landscape? So like the historic legacy that they have on that landscape. And so we originally had tried to do an occupancy model focused on wetland restoration technique. So we had ditch plugging, canopy thinning, prescribed fire, and time since those events in there. But we found what actually ended up being a better proxy was canopy cover. And so we found that it varies by species, of course. So you have your vanids, you have your true frogs like bullfrogs, who are pretty unaffected by canopy cover. So as canopy cover increases, occupancy probability increases. But for those priority species, it was pretty ubiquitous that all of them seemed to the occupancy probability declined with increasing canopy cover. And so that's something that we know already a little bit from our previous literature review and stuff. But to see it on the real landscape was really important, and to show that it might not matter as much about how you get there, but the end result needs to be that you have lower canopy cover.
Yeah, so whether you had like a fire-maintained landscape like at HOA, or you had these places where they had been doing the hack and squirt type approaches or other things, as long as that canopy cover was down, you still had higher occupancy across these species.
Yes, and the few cases where that was not true was where that variable landscape came in. And so for our rare species, like the gopher frog or the striped newts are one of my favorite examples. They are in range of all of our landscapes, so those 5 different landscapes, but we saw low occupancy or no occupancy at a lot of sites. And just because you do restorations on those sites doesn't mean that they're going to naturally recolonize those areas. And so that's where you have to take a further step and do alternative management or additional management actions like translocations or repatriations for those species. And so it's a good way to identify, like, you might have good habitat, but they're simply just not there on the landscape.
Yeah, so you had a lot of that type of error, if you will, but like, what were there certain conditions that those species generally always preferred? So yeah, you'd have good condition wetlands, but you didn't always have those species. But when you did have them, were there certain features that they always preferred? Maybe it's back to canopy cover. Did they always prefer open canopy cover? Were they, you get what I'm saying?
Yes. So let me just pick one for right now. So for striped newts, they always preferred lower canopy cover. Now, we have very few sites with striped newts still left in Georgia. And what we know about one of the sites where they're really kind of considered to be holding on and reproducing reliably every year, it has a really long to relatively high hydro period, but our occupancy model predicted that occupancy for striped newts decreases with increasing hydro period. And so that was a little bit of a surprising result, but not surprising based on the wetlands in our study. And so one of the cool things about this model is that as we get more data on occupancy, you can always add data to the model. And so we're able to adapt and learn more as we go. Distance to nearest occupied wetland was very important for all species, except for our species that are really, really common, because their occupancy was 1 across the landscape. They were in every wetland we detected. So southern cricket frogs, Pseudacris ocularis, little grass frog, they were pretty much in every place that we looked for them. Spring peepers were the same way. Leopard frogs, southern leopard frogs. You can't throw a rock and not hit a southern leopard frog in South Georgia.
How about gopher frogs? Did you do modeling with gopher frogs as well? What similar did you only find them in relatively open canopy wetlands?
Yes. So we found gopher frogs. Eva, do you know off the top of your head how many wetlands you think we found gopher frogs at least on your 2 sites?
We had 10 at HOA and 3 at Ceylon.
Eva actually might be better to talk about Gopher Frogs specifically because she ran an occupancy model on just Gopher Frogs.
Yeah, well, I was trying to do a tricky segue, and I was going to talk to you about Gopher Frogs, and then I was going to actually say how clever my segue was and transition to Eva, but that's fine. You can ruin it, Jade. So Eva, let's talk about Gopher Frogs.
Yes.
So yeah, talk to us. I guess we'll start right there with kind of occupancy modeling. You went in more focused work on the species. So why don't you go ahead and tell us about that?
Yeah, and so I had a very similar model to Jade, but just on Gopher Frogs, and so the model was able to have a little bit less uncertainty. And I also had one other method of detection. So I incorporated terrestrial surveys. And so we would take a tortoise burrow camera and stick it down a tortoise burrow and see if we saw them.
Did you find many using that technique or no?
Yes.
Really? That's cool.
I had 2 focal sites on Jade had 5. I had only 2 sites. So I did the Jones Center at Itchway and then Ceylon WMA on the coast. And so at Itchway, we had really high detections at wetlands. So we had occupancy at 10 wetlands across the site. But at Ceylon, we only detected Gopher Frogs at 3 of the wetlands. But we had like 49 terrestrial detections. So we were really successful with detecting them terrestrially at Ceylon. And so we would just scope burrows around historic or wetlands we thought that looked good. And we were really successful after the breeding season. So if we scoped in like June or July, we were just finding like handfuls of Gopher Frogs. And my model did find that the same trends that Jay was saying with canopy cover. So the Jones Center has these beautiful open canopy wetlands where Ceylon, the wetlands have become more degraded and have a higher closed canopy. So we think that's probably driving some of the lower occupancy that we're seeing on Ceylon.
Gotcha. And what were the rates again between the 2 sites?
10 wetlands on Itchway and 3 on Ceylon.
Occupied. Out of how many though? Like what's the rate, the occupancy rate?
They both have, hundreds of isolated wetlands.
That you surveyed. Okay.
No, we only surveyed. So at Itchway, we surveyed 13, I believe 13 or 14. And then Ceylon, we surveyed 14. But we decided which wetlands to survey from previous landscape scale surveys that picked out the best or historic Gopher Frog wetlands.
Gotcha. So overall, pretty low occupancy rates at both sites, but higher in the less, I wouldn't say less disturbed, but because it's more highly disturbed in a sense, but in the more like historic condition, more ecologically functioning landscape of Itchway.
And yes, but Ceylon is also naturally very different than Itchway. Like I said, it's on the coast. There's these tidally influenced streams that are disrupting the sandhills. There's maritime forests and areas of salt marsh. So it could also be more of a natural process there as well. But it is a new WMA. So I think it became a wildlife management area in like 2020 or 2021. So it doesn't have the long management history that Itchway has.
Yeah, exactly. Okay. Interesting. Well, let's stick with the Gopher Frogs. So that's kind of one major component of your thesis work. What were some of the other, you had 2 other chapters, I believe you told me, right? So there's one focused on genetics, and what was the other?
I used an individual based model.
Okay. And which do you want to start with?
We can start with the genetic one.
Perfect.
The occupancy and genetics sort of build into the individual based model. Mm-hmm. And so for my genetic chapter, we wanted to use landscape and population genetics to look at connectivity across both the properties. So like I was talking about, they're very different landscapes, and to see what features are influencing connectivity. And you can use genetics to do that by looking at level differences between different wetland populations or genetic differentiation.
So So tell us a little bit about what you found.
Okay, so this chapter used 10 microsatellites. So that's more of a population genetic based project. And so I can talk about it later, but later on we're going to use a different method. And so we found high connectivity on Ichwe and lower connectivity on Ceylon. This populations on Ceylon were highly structured. And we think that structuring is being driven by those tidally influenced streams and also degraded wetland conditions. And we did see some spatial structuring occurring on HOA, mostly driven by there's a paved road that runs through the property, and also the Itchaway-Notchaway Creek separates some of our wetlands. So that's what we think is driving genetic structure on both the properties. But we also saw evidence of low genetic diversity, especially on Ceylon.
Huh. So the paved road, that's interesting. You're talking about the paved state road, not the road within Itchaway that okay, interesting. It's still kind of interesting that's provides genetic structure there. So
I'll say that the wetlands are sort of separated like north and south, and the creek and the road separate the same wetlands. And so it could be driven by both of them, by one of them. We'd have to get samples that exist between them to be able to really tease that apart.
Yeah, I gotcha. Oh, it's interesting. So, but in general, you saw a pattern with you got higher genetic diversity at the Itchaway site, lower at Ceylon. You're seeing more structure, or in other words, like the different breeding ponds are more different from one another at Ceylon than Itchaway. Less gene flow. Interesting. And okay,
and we chose these two sites mostly because they are some of the only sites in Georgia that are known to have multiple breeding wetlands. And so, like we have other sites like Sandhills WMA where there's like one major wetland. And so we were able to use these sites to look at connectivity. But even though we're seeing lower genetic diversity, is surprising and worrisome for the populations that exist within one wetland and don't have multiple populations on a site.
Yeah. Very interesting. So tell me a little bit about, are there any other aspects of the genetics part of the study that we missed, or is that the general result?
Yeah, that's the general. So I am starting a PhD in the spring, and the first chapter of that PhD will be using a different genetic method to look at this at a more fine scale level. So more to come from that.
Okay, great. So we'll talk about that as we get towards the end. I want to hear what both of you are doing in the future, but we'll come back to that. So you're taking this, the occupancy modeling, and then you're taking the genetic information and you're developing these individual-based models. So I guess first of all, why don't you tell our listeners, like, what is an individual-based model? A lot of these people won't even be scientists. So what is an individual-based model? And then maybe get into kind of how you built a model.
Yeah, so an individual-based model is a type of simulation models, and simulation models mimic systems while allowing you to control processes and parameters, which allow you to complement or act as an alternative to field studies, especially for rare or cryptic species. And with simulation models, you can do repeated simulations that can lead to more confident inferences. It's like having multiple worlds that you're able to sample instead of in the real world, you can just sample one world. And individual-based models, individuals are organisms and they represent entities that can interact with each other and the landscape. And so you see these biological processes modeled at the individual level, and then you observe large-scale patterns occur through space and time. And so you can do them in a spatial landscape. And so the model I built follows the annual life cycle of a gopher frog and it's just on Ceylon, not Ituay. And it's parameterized by studies from our lab and other labs. And so you have a little digifrog in the landscape of Ceylon, and it follows sort of a chronological code that I wrote it. So if the model pulls weather, and then the frog will move to a breeding wetlands. It can breed, survive, create tadpoles. They can survive, become metamorphosed, and then disperse out into the landscape. And all the survival and the movement is from field studies from other labs or our lab.
Hey everybody, I just wanted to take a break and tell you guys that snakes are one of the most persecuted groups of animals in the world, but we are dedicated to changing that. The Orianne Society works every day to ensure there's a future for these amazing animals. If you care about snakes, wildlife, or wild places, and you like what you've been hearing on the Snake Talk podcast, support us today by donating on our website at www.orianne.org.
Yeah, so you're taking all kinds of data. I mean, I'm familiar with this. We've done this with various species too, but you're importing whatever movement data, and there's seasonal data about why an animal might increase its movement at a certain time. You might direct them towards certain places. You're essentially trying to create, like, you're taking what an individual frog would do in a given year, and you've got these rules and in this model, and then you put a bunch of frogs on this landscape, and you just let all the frogs do their thing on the map, if you will. And you look at whatever it might be, population level consequences or whatever your end metrics are. And so it's kind of an interesting tool. It's also an interesting tool for like simulating. I'm assuming you maybe did some of that, looking at, different scenarios for what could happen on that piece of property and what it could do to the frogs. And so I guess touch on that real quick. Did you do anything kind of simulation wise, whether that was like simulating more management? We've already heard that like open canopy is good, and you've already seen more structure in some of these things on Ceylon. So did you do any simulations as to see the effects of those?
Yes, I did a lot, and it took me forever. The model takes over 7 days to run. And so
yeah. Well, at least you weren't just sitting there listening to frogs chirp.
Yeah.
That was a joke. Go ahead.
I had an army of frogs. But one important thing about my model is each frog was assigned a genetic string. And then it's calculated from their, tadpoles are calculated from its parents every time there's a reproductive event. And so we did different simulations we'd had 2 different canopy cover layers. So one canopy cover was digitized, I think in 2019. And then we used the canopy cover from Jade's project as a separate scenario. And then we did a couple of different landscape resistance scenarios. And so we assigned different landscape types. So like pine plantation or longleaf pine or maritime forest, different values that would slow down movement. So like you would assume longleaf pine would not slow down the movement of a gopher frog, but something like salt marsh would impede movement. And so we looked at different landscape resistance values. And we ended up finding not much from the landscape resistance scenarios, and that the structure or population numbers were really driven by the different canopy covers.
At the wetland, not just...
At the
are you talking
wetland.
canopy? Okay. So it has to do with their ability to use these wetlands as breeding, not necessarily like they could in theory have a very thick canopy stretch of forest and they could move through that potentially. It was more about being successful breeding.
Yes. Yep.
Very interesting.
And
Okay.
I'll say the first thing we did with the model was make sure it worked. So that's like the first major result we got from it, was that we were able to reflect patterns we saw from our genetic study and from our occupancy study and from historic data. So like the first win was that we got the model to reflect what we were seeing in the field. But interestingly, we were only able to get those patterns if we were using the canopy cover layer from 2019. And so when I used the current canopy cover data, it was just predicting occupancy at one area of the property, which is not what we see now.
So, Eva, do you have anything else that you found out through the modeling?
Yeah, so we used the model then once we found out that it was working and validating what we were seeing in the field, to just broadly investigate the population dynamics on that WMA. And so we saw that they broadly sort of functioned as metapopulations, which are populations connected by relatively rare dispersal events. But we were more seeing source sink dynamics driven by that canopy cover. And then the next steps for the model are we're able to evaluate the effects of different management actions before deciding to invest time and resources. So Ceylon really hasn't undergone much wetland management. So we could use the model to sort of plan areas that would be most effective for improving gopher frog persistence on the property. So we could test like reducing canopy cover at a wetland and how that would improve connectivity across the landscape. Yeah. So that's the next steps for that model.
Ah, okay. Excellent. Great. Well, let's switch it back to you, Jade, and I want to talk about the other component with your thesis, and I think it has a lot to do with striped newts and has to do with also with these restored wetlands. But why don't you tell us a little bit about striped newts and what you found out?
Yeah, so the reason we decided to add a chapter about striped newts is that whenever you're doing any type of habitat management or restoration, there are a few key hypotheses that you're making. And one of those, we like to call it the field of dreams hypothesis, this like, if you build it, they will come idea. And so you're assuming that something you're changing in the plant community is going to affect that target species. And so that's what we were looking at with our occupancy model. But one of the other important assumptions in that field of dreams hypothesis is this idea that species can and will colonize restored areas without any kind of additional direct management. And so in reality, that's not usually what happens. A lot of times, especially in coastal Georgia, populations have been extirpated or they're existing at such low numbers that you start to see inbreeding depression or genetic bottlenecking. And they have these really low genetic diversities like Eva was describing. Or it may be an issue of that your restored sites are too isolated from source populations. And so one of those species that has been widely extirpated across its range is the striped newt. And the striped newt used to be present all throughout Georgia. They're still present, or all throughout the coastal plain of Georgia. They're still present in Florida, and they're doing pretty well in Florida in some parts of their range. But they're doing very, very poorly in Georgia. And we only have about one reliable breeding population of them. And I think 4 or less extant populations that we know about in Georgia. And so they've been trying to do more reintroductions or translocations. And so that's something that gets thrown around a lot. People tend to mix up those 3 terms, repatriation, reintroduction, translocation. I tend to use translocation just to mean moving animals from one place to another. And so that can be captive bred animals being moved to source wetlands where they came from, or to a new wetland, or moving individuals from one site to another. So I tend to stick with translocation, but I know there's some debate about that. And so one of the things that came out with some work by Dr. Corey Navas is that in Georgia, in the wild, striped newts tend to develop better into these patatypic adults, so they retain their juvenile traits. They'll retain these large gills and aquatic traits, and they'll tend to stay in the wetland and reproduce within the first year of their life. And so that leads to rapid recruitment. And if you're trying to reestablish a population or establish a new population, that's really important, because if you can get more newts out faster, that's great. You don't have to wait for that survival to the next year or the next year. And so we wanted to see if we could get newts to remain paedotypic after translocation. And we had a collaborator at the Atlanta Botanical Gardens, Chelsea Thomas, who was able to modify current husbandry techniques such that she could get those late stage gilled striped newts. And so juveniles that were on the larger side that were still gilled. And we wanted to know whether or not the stress of translocation would induce them into metamorphosis, either to a terrestrial adult or an eft. And so one of the other things we wanted to look at is how many newts are too many newts. And so we had this question of stocking density. We were using soft release enclosures in the wetlands so that we would be able to monitor them. Striped newts have a dismally low capture rate. And so we used these soft release enclosures that were essentially cages in the wetland to dip net for them and monitor them monthly. And we were curious how varying their stocking rate would affect competition or cannibalism and their ability to reproduce. So we assumed that if there were too few newts, there might be too few for us to see reproduction. And if there were too many newts, we expected there might be potential cannibalism of the larvae or cannibalism of each other, or just too much competition that started to fail out. And so what we found was essentially that. We also, I guess I should backtrack and get into how we monitored the newts. We received 120 newts from Atlanta Botanical Gardens, and we gave each of those newts a little tattoo. So they all got VIE, and they were individually marked, so they all had unique IDs. And we recorded all their sex, lengths, gill lengths, and total lengths, and then we put them into those soft release enclosures that I mentioned, and we dip netted those monthly. And so we added vegetation into those so that they had places to hide, but also places to lay eggs. And by month 1 of monitoring the release newts, we started to see healthy paedomorphs. So we saw those adults with big, luscious gills. And we also started to see gravid females, so females that had eggs. And then by month 2, we actually got eggs in the soft release enclosures, which is really exciting. And we just had a herp note come out cool. in herpetological review about that's the first evidence of wild reproduction by captive bred newts in the wild. And then we took those eggs back to the lab to confirm that they were striped newts, because they're so teeny tiny. They just kind of look like a cashew when you first find them, a little teeny tiny cashew. And we did hatch them out, and we were curious because they are so small when they hatch if we'd be able to detect them. But the next month we were able to dip net them up, and we found, we call them F1. So that second generation of striped newts, we found F1 individuals that were a month old. And so they were about 10 millimeters long total length.
Oh, very cool. So what did you were talking about varying densities and concerns about like their ability to breed versus cannibalism. Did you vary densities in the reintroductions, and did you see any effect of that?
Yes, so we had 2 replicates of each density, and we had a low density, medium density, and a high density. And what we found is that we had pretty high overall survival across all the enclosures, but naive survival in the high densities was lower. So our high density enclosures were about 65% survival. And then our medium and low density enclosures are about 83 to 85% survival. And for reproduction, we did not get any surviving offspring at the low densities. And I failed to mention another portion of the project. So once we got reproduction, we removed the adults from half the replicates. And so we had one replicate that was just everybody in there together, offspring and adults. And then we had another replicate of the densities that was just offspring. And then we had removed the adults. And so when we removed adults, we had higher survival of offspring. No offspring survived when adults were left in the enclosures. And so we assume this is because of larval cannibalism. And by the end of the study, we had found, we pulled the enclosures 10 months later as the wetland was starting to dry. And so we found adults that were F1 individuals who had fully sexually matured within 10 months of being released.
Interesting. So talk a little bit about maybe with both of these projects, like the application. Like these are, some of the species you're working with are incredibly rare. They're species of concern. They're threatened. And how do you think that the research projects that you did, even during your graduate work, could really help inform conservation and management of these species into the future?
So one of the important aspects of this work is adapting based on what we know. And so with the stocking density portion of the Striped Newt project, one of the things that we didn't know is how density dependence would affect their survival, their rates of cannibalism, reproduction, or the probability that they remain a petatype in that guild form. And so answering these questions can help us more effectively manage these species, especially when you're dealing with rare or threatened species. You often have a very limited number of them, or you're constrained by what you have. And so being able to get the most bang for your buck, essentially, and doing translocations effectively is key. And so being able to learn little bits as you're doing that management is really important. And so that's where that adaptive management framework comes in, where we knew we wanted to put striped newts back on the landscape. And so while we did that, we wanted to learn as we went. And so we asked questions and released them like you normally would, but we did it in an experimental framework designed to answer those specific questions.
Excellent. Well, how about you, Eva? Let's talk a little bit about kind of applications and gopher frogs, maybe the individual model. We already talked about simulations. You might talk about some of the value there, or whatever you think is important. But how's your research have application on the ground for the conservation and management of gopher frogs?
I think one of both Jade and I's favorite things about working with Dr. Mayers and Dr. Smith at the Jones Center is that our research is very meaningful. And so at Ichue, like if we have a large scientific finding, they're able to implement that on the ground. And so they already have low canopy wetlands, but if they didn't, they could implement that through their conservation team there. And then Dr. Mears has connections all over the state of Georgia. And so we've started something called the Gopher Frog Working Group. And so we just had it like last week or the week before. And so both Jade and I were able to present our findings there. And we are talking directly to the people who are on the ground doing stuff. So we were able to talk to the manager of Ceylon and talk about different plans that they can do there. And my model can be used with some of that planning. And we even talked to the state herpetologist about adapting the individual-based model to another site where we're doing active gopher frog captive rearing and releasing there. So using the model there. And then on the genetic side of things, we were able to talk about different strategies to combat some of the low diversity that we're seeing on state lands. And so it's really wonderful just to be able to actually see your work being taken into account. And it's one of my favorite things about working with Dr. Mayers is that's really important to him in the lab.
Yeah. No, I agree. That's a lot of the work I do is like that. That's some of the greatest satisfaction I get out of it is knowing how it will be used to actually conserve or manage these species. So that's great. Well, so let's talk a little bit about the future. We'll start with you, Eva, because you've already gave us a little bit of a sneak peek. But you are starting a PhD.
Yes, I am.
Yeah, tell us just a little bit about that. We don't have to go into great depth, but what are you planning on doing for your PhD, and then where do you ultimately see yourself? Is that leading you? Are you interested in academia or some other kind of career trajectory?
Yeah, so I'm staying with Dr. Mayers at UGA in Mornell for my PhD, and I sort of teased one of my first projects will be finishing up some of the Gopher Frog genetic work we do. And then the rest of it really depends on where the money goes. So we have a couple irons in the fire, and the rest of the chapters will be decided by that. It'll sort of be a hodgepodge PhD, so I won't be in one system doing one thing. I'll sort of be using the genetic skills and some of the spatial analyses skills that I have to apply to multiple different ecosystems and species.
Excellent. How about ultimate? Are you interested in academia, or are you thinking somewhere else? What are kind of your thoughts for the future?
Yeah, I would love to teach. I really get a lot of fulfillment out of working with students. One of my favorite things I've been able to do with Dr. Mears is mentor senior thesis students. And so I would love to teach at maybe a smaller university, and so an academic route for sure.
Yeah. Okay, that's great. How about you, Jade? What does the future hold? Are you planning on going on further in grad school or trying to find work? What does the future hold?
For now, a PhD is off the table for me, but you never know. Things may change. Right now, I actually just started a position as a research professional with the University of Georgia. So it's a contracted position where I'll be working on a statewide collaborative project with the Georgia Department of Transportation and Georgia DNR on a project focused on vehicle wildlife collision reduction and ecological connectivity. And so it's a culvert project with species of greatest conservation need kind of as the priority. And I'm really excited about the opportunity to get to work collaboratively with some new partners and on maybe some new species, get out of South Georgia for a while. And then in the summer, I'll be a co-instructor on the New Zealand, Australia study abroad for UGA.
That sounds like fun. So, well, that sounds great. It sounds like you guys are going to have plenty of opportunity to continue seeing and working with each other in varying degrees. So that's great. Well, I like to have all of my guests tell me a snake story. So, Jade, what's your best snake story?
That's a good question. Working at Itchaway, I have a lot of snake stories, but I think my favorite is that a technician that I had hired was from Idaho, and she was a fantastic technician, but she was so excited by southeastern herps because you just don't have as much diversity in Idaho with some of those things. And she had been looking to check off all her list of herps, and an eastern diamondback rattlesnake was one of them. And we had all these wetlands on Itchaway to survey, and we have to do them every month. And so you have to cross them off the list. Otherwise, it keeps compounding. And we had a whole group of us. Eva was there, and we were in this wetland. And all of a sudden, it just turned into torrential rain. Torrential rain, lightning, thunder. If you've ever been to that part of the state, you know how quickly lightning storms can pop up there. And so we started booking it out of the wetland. And somebody was running out of the wetland and stopped and said, "Oh my God, a diamondback." And then it was just this huddle of people in the middle of a lightning storm. And nobody cared about living or dying anymore in that moment. Everybody had to stop and see the diamondback, except for Eva. Eva was hightailing it out of there. She's, I'll see y'all suckers later. She was not going to be a French fry because of a snake and the lightning storm.
That's a great story. So your Idaho colleague was probably pretty happy.
She got to see her diamondback in the most epic lightning storm of all time.
Yeah, that's great. How about you, Eva? Tell me a snake story.
In my defense, my last field seasons were out west, and like in Colorado, they're like, book it in lightning. So I was gone. But mine it does still have to do with gopher frogs, of course. But one of our technicians and I were tracking gopher frogs at a field site in South Carolina. We had been nocturnal for about a week now, so we were a little crazy. And it was really late at night, and we were tracking down this one frog, and we could not find it. We were just looking and looking and looking, and it was our last frog before we could go to bed. And it was just moving really fast. So we would get up into it, we'd get like the beep, say you're close by, and then it would take off. And like, this is a magical frog. And then all of a sudden, this big cottonmouth comes by us with gopher frog legs sticking out of its mouth. And it kept moving. And then our transmitter beeped, and we were like, oh, there's our frog.
Oh, no. That's pretty amazing.
Able to finish up for the night.
Did you catch the snake and retrieve your transmitter?
Nope.
Did you ever get it when the snake passed it or anything like that? No.
I don't know if Andy went back and got it. She might have. But that's also the field site where someone told us that if we took a ruler and we slapped an armadillo, we could get it to do a backflip. And we never got to do that either. So
Shame.
That was random.
That's my favorite story from that site.
I was say, that's a core memory, the armadillo slapping.
Yeah.
Oh, you tried it.
We attempted to. You were supposed to get, this is a herpetologist, a biologist that was helping at that field site, said that if you got a yardstick and you slapped an armadillo on the butt just right, you could get it to do a double backflip. So we were trying to do it the whole time.
Oh, that's great. Well, I appreciate both of you coming on today. Again, just pond breeding amphibians are just have always been fascinating to me. And it was great talking to you both.
Yeah, it was great to meet you.
Thank you.
And I just wanted to tell the audience to remember, snakes are animals too, and it's a privilege to see one in the wild.